Stephanie Mariel Salvador. And I'm sorry, Stephanie, I'm not even going to try with your title. So you will have to pronounce it for us.
No worries.
But please take it away.
Of course. Is it okay if I can have access for the share screen, please? So does it look okay?
Looks great.
Okay, awesome. Thank you so much. So good evening, everyone. My name is Stephanie Salvador. I am a biochemistry major, and my senior honors project is titled Synthesis and Molecular Modeling Studies of Acetylcholine Esterase Inhibitors of Donepezil Analogs for Alzheimer's Disease. And I give great appreciation to my faculty mentor, Dr. Pesek. He is here today as well, from the Department of Chemistry and Biochemistry. Now, before I get into my research, I would like to talk about the current status of Alzheimer's disease and a little bit about Alzheimer's disease. So this figure that you see right here, it is taken from the Alzheimer's Association and studies from the National Institute of Aging. And as of 2020, Alzheimer's disease is the 6th leading cause of death in the United States, especially in adults ages over 65. And it's a little bit behind heart disease and cancers. And as of Alzheimer's disease, it is a common cause of dementia, which is associated with the inability to function in daily activities and memory loss. However, early signs of dementia can occur under the age of 65. And all of these increasing values that you see in terms of financial support and debt and deaths, it just shows that people suffering from Alzheimer's disease will continue to increase in the near future because there's actually no cure for Alzheimer's disease. There are only treatments to reduce the symptoms and progression, and I will explain that a little bit later. So one pathway for Alzheimer's disease is known as the cholinergic hypothesis, and I will explain it in this diagram shown in blue. So people that suffer from Alzheimer's disease have this enzyme known as acetylcholine esterase. It is abbreviated ACHE. And people that suffer from Alzheimer's disease, their activity of this enzyme increases. So shown in blue are neurons, and you can have neurotransmitters, which are chemical signals. And these chemical signals can travel from neuron to neuron, shown in blue, through this gap, shown in white, known as the synaptic cleft. In Alzheimer's, the neurotransmitter acetylcholine actually gets degraded in the presence of acetylcholine esterase, and it gets degraded into these two compounds shown below, acetate or acetic acid and choline. And as a result, there is a lower concentration of acetylcholine present. And the goal to reduce the symptoms of Alzheimer's is to increase your levels and concentration of acetylcholine. So the rational approach in terms of medicinal chemistry is to create a drug that can inhibit this enzyme. And that is what we have right now as a current FDA-approved drug therapeutic. And this is called Donepezil. It's shown to your right. And its prescription name is known as Aricept. But the structure and chemistry is known as Donepezil. Unfortunately, this drug can only treat mild to moderate symptoms of Alzheimer's disease. So my research involves the structure activity relationship studies of Donepezil. And what structure activity relationship studies are is that I would change one chemical structure and analyze its activity to the enzyme. So my research consists of structure activity relationship studies, specifically on site C, as you see circled in green, of Donepezil, because there's been multiple studies looked at for site A and site B. So in my scheme, I designed Donepezil analogs in the lab. I use molecular modeling on a computer to predict activity and toxicological properties. And finally, I use biological evaluation to analyze inhibition, which is currently an ongoing part of the project due to the delay because of COVID. But there are ongoing results as we speak. So in the design, you notice that there can only be one change at a time. It could be adding a chemical structure or changing the position. So in this synthesis, the goal was to keep site A and site B, shown in red and blue respectively, but only change site C, shown in green. So in this diagram, the formation of the compounds was a one-step reaction, and this began with a commercially available reagent and using a reductive amination reaction and an organic chemistry microwave synthesizer. I added various aldehyde groups, shown in the middle, to obtain 4 different analogs at moderate yields. And as you could see, the circled analogs in green are the changes in site C. And I will analyze how these changes will affect activity with the acetylcholinesterase enzyme. So upon the formation of any synthesis and compounds, they must be purified, which I used this technique known as column chromatography. And this was also coupled with thin layer chromatography to observe the various stages of purified compounds. And what's pretty cool is that this can be viewed under UV light. So that's what I'm doing in this photo right here. And the structures and purity of these analogs were verified using proton and carbon NMR spectroscopy and high resolution mass spectroscopy. And these analytical instruments are in Dan Black Hall, just to verify that the structure I made is the structure shown. So moving on to molecular modeling, I used this program in McCarthy Hall known as ICM Pro to perform docking experiments to each of my 4 compounds in a model, a computer model of the human acetylcholinesterase enzyme. One part of the docking experiment involved looking at binding interactions, and it could be depicted in a diagram, which I used to analyze if the compound can bind to a special site called a catalytic site of acetylcholinesterase enzyme. So here's one example of the results. We have A and B, and A shows donepezil within the model of acetylcholinesterase. And B is one of my compounds. I believe it's compound 3. And you can see there is an N for nitrogen and a Cl for chlorine at the site C. And the small circles surrounding each of these models, they represent the types of interactions involved in binding. And this was translated into just a list of all the interactions. And overall, my compounds all predicted similar binding interactions to how donepezil, the FDA drug treatment, binds to acetylcholinesterase enzyme. And as you could see, there is an actual overlap of the same amino acids. They're actually abbreviated with the 3 letters and its position, and which suggests that there are binding interactions to the catalytic site of my compounds. Another part that was calculated is the BBB scores, because as a drug treatment for the brain, they must cross to the blood-brain barrier, or BBB for short. And a score between 4 to 6 indicates that it can cross the blood-brain barrier. And my analogs can, with respect to the FDA drug donepezil. So finally, biological evaluation was used to test the samples and were synthesized to a sample of acetylcholinesterase. So this whole process is known as Elman's assay, and this was performed in lab, and I will break it down on what happens in this assay. So you begin with a compound called acetylthiocholine. It's shown at number 1. And you use acetylthiocholine because it is more stable and can produce a color we can see when it binds with another compound abbreviated as DTNB, which is shown at number 2. Acetylcholine is very similar. Acetylthiocholine is very similar to acetylcholine, except there's one difference in the structure. Acetylthiocholine has an S sulfur instead of O for oxygen. So what happens is acetylcholinesterase enzyme can recognize and bind to acetylthiocholine the same way as acetylcholine. And what happens is acetylthiocholine can be broken up into two compounds, acetate and thiocholine. And the one we want to focus on is thiocholine. Because at number 2, when thiocholine binds to DTMB, a reaction occurs, and one of the products produces a yellow color that you can actually see in the naked eye. But if an inhibitor is present, then the enzyme would be inhibited and not break down acetylthiocholine. And acetylthiocholine would not be able to bind to DTMB, and it would result in pretty much no color. So we use this assay to measure IC50 or inhibition activity for donepezil, and that was measured at 44 nanomolar, which means 50% of that enzyme will be inhibited at 44 nanomolar concentration, which is a really good inhibitor because the goal is to have the lowest possible amount of drug to inhibit your target enzyme. And one of the compounds that was just screened yesterday, it showed an inhibition of 512 nanomolar, which is about 10 times lower than donepezil. And what this tells us a lot about site C. Even though that this compound did not show inhibition, we can conclude from this result that the structure having 2 chlorines and 1 nitrogen is not very tolerated in the enzyme catalytic site. So in a way, we can rule out the certain chemical structures at certain positions when looking at activity with the enzyme. The screening, again, is still ongoing due to the delay of COVID, and I will have results of the other compounds, and hopefully one of them can have inhibition closely to donepezil. But overall, my research just showed the importance of site C, and if any changes of site C occur, it could either improve or destroy activity to the enzyme. And with that being said, I'm very grateful for all the invaluable skills I learned in this project, and thank you so much for listening. Let me try to exit.
Thank you, Stephanie. In a moment, we'll go to Q&A. I just want to take a moment to send a heartfelt thank you to the faculty mentors who support our students. Those of you who joined tonight, and Dr. Sines, I know couldn't be with us, but their work could not be possible without your support. So thank you so much for your generous guidance, for all the support, all the hours that you have taken to guide these students. We really appreciate it. And I know they really appreciate it too. So thank you so much to our mentors. All righty. So we have 6 screens of participants this evening, and I can't see everybody. I'm going to ask you to just unmute yourself if you'd like to ask a question to any one of the presenters, please.
I have a question for Stephanie. So looking at everything that you did in the lab, like how long did it take for you to see these results for all the 4 different components that you had?
Great question. Thank you so much. So it was for compounds. I think sometimes I wanted to shoot for more, but realistically, it was during the semester, the fall semester of 2019. And pretty much one compound from start to finish. So synthesizing it, purifying it, and making sure it's in a good yield because you want to have a good amount to test. And of course, doing the analytical tools just to verify. It took pretty much 1 day, so about 8 hours. And sometimes because they looked very similar, like the site A and site B, what we did was we kind of made a lot of just the general compound, just purifying the site A, site B part. And then pretty much you would just add your site Cs. But of course, there's always troubleshooting, but I would say each compound just a day. It wasn't consecutively, it was just kind of like when there was time.
Thank you.
Of course, thank you.